Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Michael Brewer, Director and Global Principal Consultant, Regulatory, Bioproduction Group from Thermo Fisher Scientific.
Thanks, Alex, and thanks everyone for joining. Today I'm going to focus our conversation on describing our rapid qPCR mycoplasma test and really discuss some of the considerations for implementation across multiple therapeutic modalities. I'm going to start off by talking about some of the regulatory considerations and regulatory guidance that's available on qPCR mycoplasma and nucleic acid-based mycoplasma testing methods, as well as the guidance specific to the type of samples that are being tested. So mycoplasma testing for cell culture produced in cell-based therapeutics. The challenge is mammalian cell cultures used for manufacture of therapeutics or cellular therapies must be tested for mycoplasma and be free of mycoplasma. Traditionally and in the past, the only test accepted by regulatory agencies was a 28-day culture-based test. That in the past 10 years, it's become more and more common for people to switch to nucleic acid-based testing methods for mycoplasma testing, including qPCR. The 28-day test requires specialized expertise, live control organisms. Generally, it's done by contract testing labs at a significantly high cost per sample. And in addition to the 28-day time on test, there's the logistics of shipping and getting the sample on test and processing the results and then sharing them back to the sponsor. These long testing cycles can lead to delays in lot dispositions. And even more importantly, cell or tissue therapy products may have a short shelf life, so they really require a rapid test. The alternative is to replace the traditional 28-day test with a rapid qPCR-based test. The MycoSeq assay has sensitivity that meets or exceeds the culture-based test, and this data was generated during early validation and comparability testing directly to the 28-day test. The results are available in hours, and this enables same-day lot release. So some of the top-level details on the MycoSeq qPCR method, it's optimized and it includes fully automated sample prep as one of the sample preparation options. The software is integrated and it's built for function software, which enables not only automated results analysis, but importantly, it has features that enable compliance with 21 CFR Part 11, the electronic records. The sensitivity of the assay itself is very sensitive. It detects as low as 1 to 3 genome copies per qPCR reaction. Over the years, following successful validation, submissions, and review by regulatory authorities, we have acceptance across multiple therapeutic modalities. And we support our solution with an extensive support network of experienced field application scientists offering it training on the workflow, the instrument, support on validation, design, regulatory requirements, and additional support of your regulatory submission and any potential questions that come from regulatory authorities. So in summary, this MycoSeq was designed specifically to meet the expectations for nucleic acid-based tests for mycoplasma lot release testing. And I'm going to cover some of those expectations next. The guidance I'm going to outline here is not the only guidance that's available, but it's this guidance published by the European Pharmacopoeia in July of 2007 contains the most comprehensive guidance on both sensitivity and specificity expectations, but also provides some guidance for validation study design. There's additional guidance. The USP 63 on mycoplasma testing does mention that the nucleic acid-based testing is acceptable, can be acceptable with proper validation. And, currently the European Pharmacopoeia has the 2.6.7 chapter out for comments for an updated version that will contain additional learnings on use of nucleic acid-based tests for mycoplasma. And the USP as a chapter 77 draft out for just recently closed the comment period. So there will be additional regulatory guidance upcoming. But specifically, what I want to cover here, a nucleic acid-based test for mycoplasma should be specific to mycoplasma. That is, it's inclusive to as many mycoplasma species as possible, but also does not recognize or detect non-mycoplasma species that may be related to the target region genetically. Very importantly, the limit of detection expectation, 10 colony-forming units per mil or copy equivalent. That would be genome copies, and I'll go into some details on that. And then finally, robustness, and that's typically covered during your method development as well as covered during the development of the MycoSeq kit. And a lot of the details on the robustness testing that went into the detection of the development of our solution is available in the drug master file that we can provide a letter of access to for reviews in the US and Health Canada, and can be included in a regulatory support letter for other regulatory jurisdictions. So we'll talk about the testing points that are typical. So for typical cell culture manufacturing, fed batch or perfusion-based systems, there's typically you start with thawing of a cell bank and inoculating a spinner flask, and then going into increasing sizes of bioreactors. Typically, mycoplasma is tested both during the cell banking process and then importantly in routine manufacturing at the end of production on the cell culture harvest. If you're using rapid qPCR-based methods, you can expand the testing as a risk mitigation tool to additional points. Now I'm going to cover some regulatory guidance on other therapeutic modalities, primarily cell therapy as well as gene therapy. So this is some new draft guidance that came out this year on considerations for development of chimeric antigen receptor, or CAR-T cell products. And I highlight this primarily because it provides a definition of what a CAR-T cell product is, and it is defined as a human gene therapy product, which is important because when you look into the other regulatory guidance documents that are available, they are specifically more focused on human gene therapy. And we'll highlight some of the important points related to mycoplasma testing. So mycoplasma and adventitious agents release testing is recommended on cell culture harvest material. And because alternative methods may be used or may be required, so there's guidance that rapid mycoplasma tests, including qPCR tests, be acceptable following validation. There's also specific guidance on the testing point, and that is we recommend that perform mycoplasma testing on the product at the manufacturing stage when the test is most likely to detect contamination, such as after pooling of cultures for harvest but prior to cell washing. And some other guidance around the dating period of cellular products. And highlighting that it may not be feasible to use a 28-day culture-based assay, and even recommending the use of the PCR-based method or other rapid detection methods. And of course, calling out the validation expectations around sensitivity and specificity. Um, Guidance for Japan is very similar. Safety may be assured even if virus testing or mycoplasma testing is performed on crude bulks or intermediates, providing that it is justified. This is for the reason that after the process of culturing, amplification of virus or mycoplasma is not expected or can be detected with high sensitivity. So when we look at the CAR-T manufacturing process and where mycoplasma is tested, and typically and per the regulatory guidance, testing takes place at the end of the cell expansion process and prior to washing, processing the cells for final dosage form. And some additional guidance I'll touch on that's in the draft document that came out in January of this year is that vector safety testing, this would be viral vectors that are used as part of the CAR-T process, but also viral vectors that could be used for gene therapy products. They're required to be tested for mycoplasma. Then I want to cover where the test point that process is, and much like monoclonal antibody and cell culture manufacturing, when mycoplasma testing for viral vector manufacturing takes place at the end of cell culture process and prior to forward processing. So now I'm going to cover some real details on the MycoSeq mycoplasma detection system. As I said earlier, it's designed for lot release testing in a GMP environment, designed to meet or exceed the guidance in EP 2.6.7 from 2007, optimized sample testing or applications, software that enables 21 CFR Part 11 compliance. We offer instrument and software validation services in our global support network. The important part here is the MycoSeq system enables high confidence in your testing results. It's sensitive. It enables validation at levels at or below 10 genome copies or 10 colony forming units per mil of test sample. Specific to mycoplasma, no detection of off-target organisms. We use objective multi-parameter results interpretation, which I'll detail later. Has full workflow controls, including a patented discriminatory positive control for even higher level of confidence in both your negative or positive test result. And again, following validation, regulatory filing, and review, our customers received regulatory acceptance to use MycoSeq for lot release testing applications across multiple therapeutic modalities, including cell culture manufacturing, cellular therapy, and tissue therapy. It's a complete sample to answer system, and the system illustrated here includes the optional automated sample prep device, the Automate Express, combined with the qPCR assay and master mixes, the instruments, either the Quant Studio 5 or 7500 fast real-time PCR systems, and the AccuSeq software. In a little more detail on the automated nucleic acid extraction system, this is ideal for testing situations where multiple samples are processed in parallel, which can be common to modern commercial cell therapy manufacturing. It's fully automated for the entire workflow. From the sample is added to the cartridge, and at the end of the extraction procedure, the eluted nucleic acid ready for qPCR analysis is delivered in a separate tube and ready to go to the qPCR reaction plate. I want to cover the components of the MycoSeq assay sensitivity because it's not only just the qPCR assay. There's really three primary components. That is a background reduction in mycoplasma concentrations. We have large volume protocols for a variety of different test samples. And importantly, for cell-containing samples, our protocol enables accounting for both free mycoplasma as well as potentially cell-associated mycoplasma. We follow that with a sample prep based on magnetic beads that enables very high percentage nucleic acid recovery. And then moving to the detection stage, detection using the highest sensitivity qPCR assay. The assay is designed as a multi-primer based detection of mycoplasma in a single reaction. We incorporate a proprietary discriminatory positive control. It's sensitive down to 1 to 3 genome copies per qPCR reaction, and again, specific to mycoplasma. All these features enable the highest sensitivity mycoplasma detection. I really want to cover in detail the rationale for using higher test sample volume, especially for the lot release testing application. So typically for the lot release in our highest sensitivity protocol, we call for 10 mils or more of starting sample volume. This is eluded with our standard sample prep protocol at 100 microliters, and we test per qPCR reaction 10 microliters per sample. Using the LOD guidance of 10 genome copies or CFU per mil or per reaction, we are processing and testing the equivalent of 1 mil of the starting test sample in a single qPCR reaction, enabling that bottom line sensitivity of 10 GC or CFU per mil. If you process lower test sample volume to start, you get a subsequent decrease in sensitivity. And while these protocols are perfectly appropriate for in-process testing, where you're testing throughout the manufacturing process, the sensitivity that needs to be obtained at release is the 10 GC or CFU per mil. And just one more slide on that. Do 10 and 1 mil starting sample volumes enable detection at the same LOD? Well, if you start with a 10 mil sample of mycoplasma at 10 CFU per mil, process 1 mil per qPCR, you end up with a sensitivity or a final concentration of per qPCR of 10 CFU or GC per reaction. Conversely, if you start with only 1 mil sample of that 10 CFU per mil level of mycoplasma, 1 mil of starting sample, testing the equivalent of 0.1 mil per qPCR, your final concentration per qPCR reaction be 1 CFU or 1 GC per mil. And although our assay does enable detection at that level, due to just Poisson distribution of a normal distribution in a sample at that low a concentration, you would not expect a detection event in every qPCR reaction if you were starting with the lower sample volume. Now on to the performance of MicaSeq. But first I want to cover the results interpretation and acceptance criteria. As I said earlier, we use a multi-primer based multiplex PCR for the amplification. The detection with MicaSeq is based on cyber green dye, which is a dye that becomes fluorescent when it's intercalated into double-stranded DNA, when that double-stranded DNA is created during the PCR amplification of the DNA. So we use 3 parameters for analysis of results. The cycle at threshold, which is a quantitative parameter. It's a measure of the target DNA level at the beginning of the PCR reaction. We have acceptance criteria for routine testing is typically CT less than or equal to 36. We also incorporate an analysis of the amplicons created during the 40 cycles of qPCR, and that's the derivative value, which is a measure of the specific amplicon quantity that's generated. And we have an acceptance criteria on the measurement of that curve and the melting temperature. This is an assessment of amplicon size and base composition, which is known for mycoplasma using this primer set. And that criteria is the amplicon should melt between 75 and 81 degrees. Importantly, we use all 3 of these criteria to make a positive negative call. So this is what the raw data looks like. In the left-hand box here, we see the amplification plots that are generated in testing a 10-fold dilution series of purified Mycoplasma arginini DNA with a range here of 100,000 copies per qPCR reaction all the way down to 1 genome copy per qPCR reaction. So we can see amplification plot indicating detection crossing, a CT value of about 34. And then the MELT analysis of the triplicate reactions at 1 genome copy per reaction. So seeing a robust melting curve for the amplicon with a melting temperature of 80 degrees. And importantly, we've confirmed this LOD with multiple independent DNA stocks. Next, I want to talk about our discriminatory positive extraction control. So positive control DNA made with Mycoplasma amplicon modified to have a melting temperature, or TM, outside the range of Mycoplasma amplicons. It allows an additional level of confirmation of a positive test result. That is, your test, for Mycoplasma, the melting temperature should be between 75 and 81 degrees. For our discriminatory control, the amplicon melts at a higher temperature at 84 to 85 degrees. This enables discrimination between true Mycoplasma and our assay control. So there's no risk of an inadvertent contamination of your test sample with control leading to a conclusion of false positive or positive for Mycoplasma. It's easy to tell the difference between control and Mycoplasma. This also enables a workflow that you can do extraction and control spike and recovery, so confirmation of the total workflow during your routine testing. I want to show you some results from validation and also discuss how the quantitative parameter, the CT, can be used in validation studies as well as in routine testing. But here what we're illustrating is comparison of CT data from multiple validation studies, and these are testing using our lot release protocol at either 10 genome copies per mil, that's with purified DNA spike, or 10 CFU per mil, that's spiking with live Mycoplasma that's been carefully prepared to ensure a close relationship between genome copy and CFU. I'll detail that in a couple slides. And here's a second validation study. This was done slightly below the 10 CFU per mil expectation at 8 genome copies per mil or 8 CFU per mil. We can see in replicate samples analyzed, 100% positive results were obtained in these studies. But also looking at the CT values, you could see the CT values were very similar for both within the study between genome copy and CFU spiking and between independent studies. And this is important and can be helpful to regulators when they're reviewing your validation information. Next, I'm going to talk about a case study where, again, the CT value can be a very powerful tool in analysis. In this case study, a production bioreactor was contaminated with Mycoplasma. Interestingly, in this situation, the production bioreactor, all operating parameters were within normal control limits at harvest. Not using the 28-day Mycoplasma test, a result was obtained as positive. The positive organism was identified by 16S ribosomal RNA sequencing. Used microseq ID system. It was identified as E. coli plasmaloid laui. And of course, an equipment and facility decontamination was initiated, as well as a root cause investigation. A part of this root cause investigation utilized Mycoseq testing for some reserve and process bioreactor samples. So we can look at this production bioreactor. It was a 14-day process. It was fed on both day 3 and day 9. And then some reserve samples from these various time points were tested with Mycoseq at day 5 and day 7. There was no detection of Mycoplasma. At day 9, a second feed was introduced. Then on day 11, the Mycoseq result was positive. It had a relatively high CT of 34.3. We're estimating about 60 CFU per mil level of Aoade-Lowy. By day 13, the CT had dropped to 26, and enabling an estimate of about 20,000 CFU per mil. And the next day, the CT had dropped another 12 CT up to a level of estimated at 800,000 CFU per mil. So the outcome and conclusions. So the first Myco-Seq qPCR positive was at day 11. The CT levels were used to estimate the level of contamination, and then decreasing CT values at day 13, day 14 confirmed the viability and growth rate. Myco-Seq was also used to assess impact of contamination on the downstream processing equipment, and finally, included a recommendation that Myco-Seq qPCR pre-harvest testing be applied as a conditional release test going forward. So you can see an accurate and powerful tool. Now I'm going to move on to considerations in analytical testing method qualification and validation for Myco-Seq. So first, use of a qPCR detection or nucleic acid-based test for mycoplasma detection. It's important to understand the analyte and the relationship between a colony-forming unit, that is a mycoplasma cell that could be counted as a growing colony on an agar plate, and a genome copy. So a mycoplasma cell, illustrated here in pink, contains one chromosome. It's circular. That would be one genome copy per cell. The Myco-Seq assay targets the 16S ribosomal RNA gene that is present at least one copy in all Mycoplasma species, but some species do have two copies of the target gene. So ideally, one genome copy would equal one CFU, but that must be verified. If this ratio changes because the sample or stock of Mycoplasma cells contain DNA that do not grow and represent as a CFU, sensitivity of the nucleic acid-based tests may be misleading, and this is especially important in validation. And here's on this next slide, we'll go into the details of that. This is data showing Mycoplasma in culture. In the diamonds here is a measure of CF or genome copies per mil measured with qPCR, and then the gray boxes are a measure of viable CFU as measured by agar plating and counting. You can see the Mycoplasma can reach, quickly go into log phase growth, replicating quite rapidly. But at some point they will reach a stationary phase and then enter a death phase. So the CFU count starts to decrease while the DNA is measured by qPCR is staying relatively stable. And what happens there is that creates an imbalance from a very close ratio between genome copy per CFU to an imbalance where there's excess genome copy to the CFU titer. So again, your LOD results can be misleading if you're using stocks with a high GC to CFU ratio. Myco-Seq, importantly, enables detection of 10 CFU at 1:1 GC to CFU ratio, and also enables detection at Mycoplasma DNA spiked at 10 genome copy per mil. We also want to talk about some lessons we've learned over the years and explain the rationale for LOD testing with purified Mycoplasma DNA. The analyte detected with a DNA-based detection method is Mycoplasma genomic DNA. It's not a Mycoplasma cell or colony-forming unit, although it would only be expected that Mycoplasma DNA would be present if there were Mycoplasma cells in the test sample. It's well established that Mycoplasma genomic DNA can be purified and very accurately quantitated, often using orthogonal methods to increase the confidence in the quantitation. The stability of purified DNA stocks has been demonstrated. And as you saw in the previous slide, the preparation of accurately quantitated Mycoplasma stocks can present challenges, and these include variability of genome copy to CFU, as discussed, accuracy of CSU titer due to clumping or aggregation, especially with some Mycoplasma species, but either of these or both can combine to lead to misleading results. Stability of Mycoplasma stocks can be difficult to maintain even when stored at -80. So if you're using live Mycoplasma to avoid any potential degradation leading to a misleading result, we recommend titering immediately prior to use and validation testing. And that's both counting genome copies with qPCR as well as plate culture if you have the ability to do that. And then importantly, and what we have found in many of our customer sites, that use of live Mycoplasma presents an unacceptable risk, so it's just not allowed. So doing LOD validation with pure DNA does not present a risk and can be used. And finally, if concentration of Mycoplasma by centrifugation of the test sample is part of the sample prep, which we recommend for these 10 mil or higher volumes, recovery of Mycoplasma from liquid samples, including cell culture by centrifugation, is proven and widely published. And here's an example of a validation study plan that's fairly typical of our customers that have applied. Testing for specificity, no detection of non-Mycoplasma bacteria, and this is very well established in the data, and the drug master file may be acceptable for a sponsor to avoid doing off-target testing. But part 2 is the important part: no interference from the test sample matrices that are being validated for MycoSeq, which is also addressed via spike recovery in LOD.1, but that's the important part of specificity that's confirmed during validation. For LOD part 1, for each Mycoplasma species, including in the study, we recommend performing extraction of 6 samples with 100 copies of 10 genome copy per mil of purified Mycoplasma DNA spiked in the lysate from a 10 mil sample. Run 4 qPCR replicate reactions per extraction, so that will give you 24 total results, which can enable a statistical analysis of those results to ensure detection at or above 95% of the test samples. Additionally, an LOD part 2 can be performed. This can be optional or may be omitted. We do recommend discussing your validation plan with your regulators. But for each Mycoplasma species included, you could also do a second LOD arm with 6 samples spiked with 100 CFU, 10 CFU per mil of live Mycoplasma spiked directly into the 10 mil samples, and run 4 PCR reactions per extraction, again generating 24 total results to enable the analysis of the cutoff of 95% or more of the positive detection. Quickly, I'll go over the results of our external validation. And this is the specificity part 1, that is qPCR analysis of both host cell genomic DNA, because typically the cell culture sample will have host cell DNA in it, as well as microorganisms that are related to Mycoplasma at the genetic level. You can see in this triplicate reactions analyzed for all these species, there was no detection of the closely related bacterial or common host cell species. This was tested at 10,000 genome copies per reaction, so significantly high level. Second, the summary of the LOD using purified Mycoplasma DNA. Again, using a study design as outlined on the previous slides. We tested 10 Mycoplasma species, 24 total tests per species. 100% were positive. We also got the obtained CT values, which could be analyzed for consistency, especially one of the features of qPCR assays, or any analytical method for that matter, as you approach the lowest limit of detection capability of the method, generally start to see a higher level of variability in the test results or the replicate results. You can see here that the results were very, very consistent, a low standard deviation and a low percent CV, indicating confident detection, not approaching the lowest limit of detection capability of the method. Uh, and and again, 100% of the replicates testing positive at the specified LOD. So in summary, Mycoplasma, it's a real-time qPCR assay specifically designed and validated to meet lot release testing requirements. Sensitive, specific, objective acceptance criteria using all three components. The unique discriminatory positive control to enable differentiation between control and Mycoplasma test article. We also include other relevant assay controls, such as a no template control, a positive control, as well as a control to assess for any qPCR inhibition, which is very important because typically your test sample would be negative. You want to have a control that ensures that negative result is not due to PCR inhibition. And our comprehensive support, drug master file in place with the FDA and Health Canada, specialized field application scientist team globally, expertise in validation study design and regulatory support, and our equipment validation services. So with that, I want to just review our successful track record of validation and implementation of Mycoplasma for lot release testing and commercial processes. So across multiple therapeutic modalities, cell, gene, tissue therapy, monoclonal antibody, recombinant protein, as well as vaccine, our Mycoplasma has been implemented in more than 40 commercial processes and is in validation or has been qualified for use in more than 30 clinical stage processes in regulatory jurisdictions that have accepted the use of Mycoplasma is global. It's designed for purpose, confirmed in validation, accepted by regulatory agencies. It's unique that we do have 2 patents on Mycoplasma as well, both for the detection approach with the multiplex primer approach, as well as discriminatory positive extraction control. And with that, I will be happy to take any questions.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, what do you recommend as the next steps following a positive result from a test for Mycoplasma?
Yeah, and that's a good question because when you get a positive result outside of validation, obviously this is an unexpected result. It's an out of specification result, and you're going to want to learn more about that. So one, I will look at the CT value and, are you near the limit of detection, or is it a CT level that indicates, medium or high level contamination? I also look at, of course, the milk parameters. Secondly, especially if there's a significant level or a CT below about CT of 30, my recommendation, my past practice is to immediately put some of the extracted DNA into the a 16S-based sequencing identification workflow, so such as microseq ID, where you amplify the 16S target gene and sequence it, so you can use a database comparison to identify the species causing the contamination. And then often we incorporate qPCR testing to understand the scope of the contamination. How is it contained in one bioreactor, or is it in multiple points in your process, or even impacting multiple lots? So those are my suggestions for follow-up on a positive test.
And can you tell us more about what you said about confidence in test results?
Yeah, and again, a positive result in routine testing is going to be, it's an out of specification. You're going to have to make a decision on that, what to do with the fate of this test sample or manufacturing process. So again, it's following the steps I described previously. Or in some cases, do you need to do some follow-up testing to confirm that? Again, rapid testing enables, fast follow-up. So yeah, so you want to see the three parameters. You want to see a CT value, and you want to use that CT value to kind of estimate the level of contaminant that's been detected. And then with MycoSeq, you want to confirm, visually that the melt parameters are within the expected range.
All right, and how can sample matrices interfere with fluorescence?
Yeah, that's an excellent question as well, because certainly with a cybergreen-based detection method, testing a complex cell-containing sample, there can be potentially nucleic acid that could absorb the cybergreen dye and become fluorescent. So we've optimized procedures to treat the samples or pre-treat the sample both with nuclease to reduce the host cell nucleic acid level, as as well as proteases to break up complexes of degraded mammalian cells that also contain nucleic acid that could co-purify with the mycoplasma during centrifugation. So background treatment and background reduction is an important step. Then, you know, during your qPCR analysis, of course, utilizing the inhibition control to ensure that a negative result is not due to qPCR inhibition. And we can use the CT values to assign objective results acceptance criteria based on CT value for the inhibition control.
All right, well, it looks like we're running a bit short on time, so if we don't get to your question, we'll be passing them along to Mike, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, how many samples can be tested in one assay with MycoSeq?
Yeah, and that's a good question as well, especially as newer therapeutic modalities have emerged, such as cell therapy, where you may be processing multiple test samples in one reaction. So we use 96 well plate-based analysis for the MycoSeq test. So potentially, including the relevant controls for both the assay as well as the samples, you could process 44 or 45 samples in parallel. Typically, that's nobody has that level of throughput to require it, but using the 96 well format, it does enable testing a high number of test samples in parallel.
Okay, thank you very much, Mike. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.